It's great to be here. Thanks again to CED for inviting us back to give everybody an update on AgBiome. Some of you may recall that AgBiome is an ag biotech focused company. We have a large platform based on the agricultural microbiome space, a pipeline of products for agriculture that we are developing, and a very fast pace of discovery and time to market. We have a great investor syndicate, including several strategic investors, Monsanto, Syngenta, Novozymes, and our platform, as I said, is based on the crop microbiome, which means we have over 45,000 novel sequenced strains in our collection. We screen those for activity against pests and diseases, and then develop those either into biopesticides or traits for transgenic crops. That's a fully integrated, data-driven platform, and we think it's unique in the industry. We think our team is also unique. A majority of our folks have actually been involved in some agricultural product successes. Many of these are still on the market, everywhere from chemicals, trait products, biological pesticides, even animal health products. And we have a unique organizational structure also, which is non-hierarchical. We don't have a management hierarchy or a supervisory hierarchy, I guess is the right word for it. And we think this is also a significant advantage as far as unlocking some of our success, and I'll come back to that. So what we've realized recently, and yeah, I thought I could come in and talk to you guys about our product pipeline and our biopesticide that's about to launch, but I think what I decided to talk about instead will be more interesting to this audience. Our core IP is that collection of microbes, and we've discovered that it has utility, of course, outside of crop protection. And so while that will encompass pharmaceuticals and industrial biotechnology, we've also found that it has utility in gene editing, and that's a hot topic right now. You've heard a couple of talks about that this morning already. But based on our collection of strains and unique genes, we found that this is going to also provide us with a significant business opportunity. And we have spun out a subsidiary called LifeEdit, and you will be able to learn more about that if you come see us at our table in the innovation room. The other thing that's really interesting about this is that the discovery of the novel gene editing systems that are in our collection was driven by an intern. And this is where our unique organizational structure really comes into play. The intern had complete freedom to look into this topic, discover a lot of novel gene editing tools in our collection, and we decided based on her work to go ahead and set up this subsidiary. So we think that's a significant advantage. We were able to make all of this happen in a very short period of time based on her work. And we're focusing first off on therapeutics and also on agriculture. Obviously, we know a lot about ag, so that's something interesting to us. We have over 3,000 RNA guided systems identified so far. And you'll understand, having followed this field, that that's a significant advantage. There are about 10 validated tools for gene editing currently known. Type 2 systems amount to about 200, and we have over 800 plus of those just in our collection alone. So this is going to allow us to do things like overcome immunogenicity concerns, edit any gene at any position, introduce genes seamlessly. We can adjust the application method depending on the type of protein that's found or the target that's desired. So we've only searched about 60% of our collection, and you can see that the number of genomes that we've identified is already a significant advance over the number that were already known. And this will allow us to look at different targets within the nucleic acids. You can see some of these are targeting double-stranded DNA, some single-stranded, some staggered cuts. So this will allow us, again, to target pretty much any system that is of interest for editing in the genome. And of course, this is some new research that was published last year, Nature Reviews Microbiology, showing the type 5 uncharacterized systems. And we've identified over 13,000 of these by genome sequence similarity in our collection. So while these are still uncharacterized, they're not yet validated, we think there's a huge opportunity here, a challenge also, of course, but a huge opportunity for novel gene editing systems. And this is a real area where a large collection like ours makes a big difference. We can search the collection for these kinds of things and find what we're looking for rather than having to develop it synthetically. So the company is actively looking for partnerships right now. We are not intending for this to become a reagent supply or a one-off sale. We are interested in any kind of partnership, licensing arrangements within the fields that we're interested in, and then eventually moving into developing our own products. We've assembled a great team to work on this already. A CEO of the subsidiary is Mark Moore, who, among other fantastic experience in his background, was one of the co-founders of Gliologics, and some great scientific talent, including Allie, who is our intern who first started searching the collection for these tools, and she is here with me today, so you can talk to her as well. And she came from Dr. Barangou's lab at NC State. So that's what we have to update you with today, and really hopeful that this will generate some interest within the local community for an additional approach to gene editing and some novel tools from our collection. Thank you.